This is definitely a passion of mine, as well as a number of other people at Google. The whole aspect of how we perceive the world and cognition being a big part of that. So without further ado, I'm just going to let Mike and Em speak eloquently to their work.
Hello, everyone. So my name is Michael Goard, and I am a recent graduate of the neuroscience program at UC Berkeley. And so today I'm going to talk a little bit about the neural circuitry underlying the perception of the outside world. And so over the last 5 years, I've been studying how the visual cortex of rodents, mice and rats, encodes information about visual scenes that they're seeing. And so on the right here, I have a schematic of a rat brain. And basically what happens is images coming into the eye get transferred through a relay in the thalamus to this area in beige up in the upper right called the visual cortex. And what we can do as neuroscientists is drop an electrode into his visual cortex and record the electrical activity of individual neurons and see how they are responding to the visual scenes that the rats are seeing. So this is usually how I start the talk, and then I spend an hour going into all the detail and the data that I've collected over the last 5 years. But today I'm going to do something a little different. I'm going to take a big step back and address what I think is a very important question, which sometimes as scientists we don't get a chance to address. And it's the question of who cares? Why are we interested in the neocortex? I mean, first of all, what is the neocortex? Why do we care about the way in which it encodes sensory information? And if we can figure out how it works, what will we get out of it? So first question, what is the neocortex? So shown here is a human brain. So if you've seen it, you kind of know on the outside it's a big kind of wrinkly mess. Looks like a walnut with kind of a division down the middle. So we've just kind of popped it in half. And we're seeing the inside. And you'll see that there's this kind of wrinkly rind on the outside. That's the neocortex. And on the inside, there's some smoother structures, which are the basal ganglia, the thalamus, the brainstem, and this kind of cauliflower-looking thing. This is the cerebellum. And so all this stuff in the inside I'm going to refer to collectively as the quote-unquote old brain. The neocortex is wrinkly stuff on the outside. So this comes from the Latin cortex, meaning rind or bark, because it kind of envelops the older structures, and neo because it's new. So this is evolutionarily a recent invention. It's particular only to mammals, and it's only grown large relatively recently. So why are we interested in this? So I'm going to kind of lay out a few brains for you. So starting at the upper left, we have a shark brain. Now, the shark brain is pretty much exclusively the kind of old brain structure, so the middle parts that I showed you in the last slide. And there's only a very small part that's kind of cortex-like that's involved in sensory processing. Now we go one down. The next one over is a rat brain. So this one is kind of mostly the old brain, but it's got a single sheet of the neocortex covering it. One more down, we have a cat brain. So here you can see that we're starting to get more of this kind of neocortical covering. And you can see it's also starting to get kind of wrinkled. So the neocortex is a sheet, and it has kind of a fixed width. And so one way of kind of squeezing more of it in a fixed volume is by kind of convoluting it and wrinkling it up, so you get more surface area per volume. And this is kind of carried to a logical extreme in the human brain, which you can see is a very kind of complex mass of contortions, and it's almost completely taken up by this neocortex. So while, say, in the rat, it's only about 25% of the total volume, in the human, it's over 75% of the human brain is neocortex. And so it seems to be doing something interesting. And in fact, if you kind of look at the size of the old brain compared to the size of the creature in general, you find a pretty kind of proportional increase. The bigger the creature, the bigger the kind of old brain. But the neocortex, you get this kind of unusual overexpression of it in certain species. So humans are probably the best example of this. We have 6 or 7 times what you kind of expect given our body weight. Something like a monkey or a dolphin might have 4 or 5 times as much. A dog or a cat would have a little bit less. And so what we see is kind of the more neocortex has, the more kind of complex behavior and cognition you're getting from the animal. So this is kind of why we think it's interesting. So this is just kind of summing everything up in broad strokes. So the old brain is kind of responsible for reactions to stimuli, the automated motor sequences, control of your peripheral nervous system, whether it be temperature regulation, circadian rhythms, whether you're awake or asleep, arousal. Also emotion. We tend to think of emotion very Shakespearean terms, but it's actually very much part of the old brain. And, this makes sense. Even very simple organisms need to have emotional valence. That tasty morsel over there needs to be good, and that thing with the big teeth needs to be bad. Now, the neocortex, so the old brain kind of gives you everything you need for survival. If you see something that's food, you can bite it, you can reproduce, you can run away. But the neocortex is involved in kind of the more complex cognition that we associate with intelligence. So complex sensory processing. So not just kind of seeing something and reacting to it, but being able to understand, that it's an object or what its uses might be, or that it's an individual and a certain individual. Motor planning, the idea that you don't just kind of react to something, but we might plan to do something later on today and we'll actually do it. Abstract reasoning, theory of mind, the idea that we can understand what other individuals might be doing. And all of these, by the way, we tend to kind of think abstract reasoning and theory of mind are things that only humans do, but there's abundant evidence that other animals do it as well. Consciousness is kind of a stickier one, but we do know that the neocortex is somehow involved. There's this interesting phenomenon called blind sight, which is that if a person has damage to the back of their brain where the visual part of the neocortex is, and then you kind of flash a light in a certain location, ask them to point at it. They can point directly to it, but they have no conscious percept of the light. They'll tell you that they can't see it at all. So it seems like consciousness is somehow kind of subserved by the neocortex, so we're not sure how. So in order to kind of subserve all these different functions, the neocortex must be this very kind of complicated, heterogeneous structure, right? And the answer is kind of yes and no. So you may have seen these maps before. So this is, the human neocortex, and different areas kind of correspond to what they process. So in the back here, you have the visual areas, and in the middle, you have the sensory and motor areas, and the speech areas, et cetera, et cetera. And this is all true, but it turns out that the cortical structure is actually remarkably homogeneous kind of across the whole neocortical expanse. And what seems to kind of define these different areas is not some difference in the architecture of the...
Okay, great. So like you said, you had gone from,
Could all of these sites please mute their audio? Thank you. Thank you so much.
Thank you. So what seems to differentiate these different regions is not so much the structure of the neocortex in that region, but rather the inputs that are coming into it. So what does the structure of this neocortical kind of circuit look like? So here we have a picture of a human brain. So this darkened kind of band here is the neocortical sheet that's all convoluted. And if you just kind of take a section of it and then turn it upright, so on the top we basically have towards the outside of the brain, and on the bottom we have towards the middle of the brain. You see this kind of layered structure. So the cells have kind of different densities and they're different sizes in the different layers. And this is very kind of stereotypical no matter which chunk of cortex you grab. So whether it be a visual part or a part up here involved in abstract reasoning, it really kind of looks the same. And so what's going on here? So you can see these little balls with kind of little lines coming off of them. So each one of those is a single neuron. And you can see the neuron kind of blown up here in the upper right. Unfortunately, this image hasn't totally come out. But so the kind of ball in the middle is the cell body. And then there's a bunch of processes coming off of it in kind of a tree-like structure. Those are the dendrites of the neuron. And that's where it collects all its inputs from other cells. And if it gets enough input at any one point in time, it gives off this little blip of electrical activity called an action potential, and will send that activity to other cells.
Do people always have 6 layers and kids with without?
Yeah, so the neocortex is always 6-layered, everywhere in the brain, everywhere in development.
Is that layering the big difference between the neocortex and the old brain?
Yeah, so the old brain doesn't have any layers. It tends to be kind of discrete nuclei. There are kind of primitive areas of cortex, like the archaic cortex or the hippocampus, which have a few, like 3 layers, for example, or some different number. But the 6-layered structures is kind of characteristic of the neocortex. And there's some other commonalities that I'll talk about. Also, I should emphasize that, so I'm being a bit of a neocortical chauvinist here and kind of talking about the neocortex as the only kind of way of kind of getting intelligence, but there's actually at least one other solution that we know of. So if you look at bird brains, they evolved very separately from us. If you remember your Jurassic Park, they diverged sometime around the dinosaurs. They actually have a different kind of structure covering their brain called the neopallidum. And it has a lot of commonalities with the neocortex. It's a layered structure. It doesn't have 6 layers, I don't believe. So there's some similarities, but essentially it's a different solution to doing the kind of processing that the neocortex does. And it turns out birds are actually quite intelligent, capable of all kinds of complex and interesting behaviors and sensation and creating songs, that sort of thing. But back to what I was talking about. So these neurons here, they're all kind of wired to each other. And neurons are interesting because they basically kind of collect inputs from other cells and then decide to either give off this action potential, this little blip of electrical activity, which it sends to its partners, or it doesn't. So you can almost think of it as like the transistor in the neural circuit. And this neural circuit kind of wires up in such a way to perform some kind of computation. So, is this really kind of a universal circuit? So there were some experiments to address this about 20 years ago, in which they took ferrets very early in development. And so normally you have inputs from the eye go through a relay. You can see it there, it's labeled the LGN, which then goes over to the visual cortex. And you have inputs from the ear go through a different relay called the MGN, and then over to the auditory cortex. So what they did is they actually ablated the input from the ear and kind of induced the inputs from the eye to go over into the auditory relay, which then went over to the auditory cortex. And what they found was very interesting. The auditory cortex, or the quote-unquote auditory cortex, now had visual responses. And the visual responses actually look very similar to what people see in the visual cortex. So it seems, at least early in development, there's a fair amount of universality in these circuits. Now, later in development, they kind of specialize for whatever inputs they're dealing with, and they become kind of less, you can't just kind of mix and match them in the same way. So we've, neuroscientists have been trying to figure out kind of what is this circuit and what is it doing. And there's been a lot of work on this, and we've made some progress, even though we're not there yet. And I've kind of quickly schematized kind of the general connections you see. And I'm not going to go into a lot of detail here. But basically, you have inputs coming in from lower regions of the cortex. So this can either be from the sensory periphery. So let's talk about visual cortex as an example. So this could be from the eye, or you also have pieces of cortex stacked on top of each other. So you have the eye, and then the primary visual cortex, and then higher regions of visual cortex. So as input, if you're in a higher region of visual cortex, it could be from the lower area of visual cortex or from the eye. You also have inputs from higher regions going down, and then there's some processing that happens in the circuit, and then it spits out inputs both up the hierarchy and down the hierarchy. And these hierarchies can be quite complex. So neuroscientists like to show this slide because it makes what we do look terribly complicated, but we don't actually know much of what's going on here. But so basically each one of these blocks kind of represents one or more of these kind of cortical circuits all in parallel. And as you can see, they're kind of stacked on top of each other. And what we do know is kind of as you go up this hierarchy, you get more and more complex representations of the stimulus. So the visual stimulus going in here, these first levels, basically what you see are the neurons respond to small kind of dots of light in a certain place in the visual field. Next level up, you see they respond to lines kind of moving across a certain area of the visual field. Up here, you get kind of more complex contours, and they also start to take up a bigger and bigger portion of the visual. So up here, they'll respond to a pretty big area, and they respond to some kind of complex contour. And at the very top, you get these very kind of abstract responses. You can get responses to, only certain objects, responses only to people or faces, or even certain people in some cases. And in these cases, it usually takes up the entire visual field. So, say it's a person-selective neuron. If a person is anywhere in the visual field, it will be active. And this property is very interesting because it's actually very hard to get. If any of you have ever worked on computer vision problems, it's almost impossible to program a computer to be able to recognize an object. And that's because there's infinite kind of ways that that object can present itself. If I have this remote, I can turn it around, and depending on how I present it to you, if I bring it closer to you, it'll be bigger on your retina. If I bring it back, it'll be farther away. And yet, no matter what I do, you can recognize it. But trying to find some kind of archetypical feature which a computer could extract and say, okay, that's a remote, is very difficult. So what seems to be happening is the visual cortex, we have this circuit that's kind of combining information from higher regions and from lower regions, and it's allowing it to do some kind of useful, informative task. So some of you may have seen this already. It's kind of a famous example, but if you haven't, I want you to try to figure out what is in this scene. And now I'm going to give you a hint if you haven't figured it out yet, which if you haven't seen it, you probably haven't. It's of a Dalmatian. There's a Dalmatian somewhere in the scene. So some of you are probably seeing it now, hopefully. If not, there's a head in the front and just kind of tracing the contour. So now, even if I take it away, everyone sees the Dalmatian, right? And so what's happening here? So you're getting an ambiguous kind of not very clear input. And once you have, the higher parts of your brain are kind of sending back useful information that, there's a Dalmatian in the scene, then you can kind of figure out where it is and what's going on. And this is actually very interesting because I didn't tell you much about the Dalmatian. I didn't say which way it was facing. I didn't say whether it was big or small or whether it was, down on the ground or howling at the moon. All I said is that there was a Dalmatian, and yet somehow your top-down inputs kind of informed your kind of lower processing regions to kind of constrain the possibility of what you could look for in order to help you find it. And this is what I think, and at this point I'm kind of leaving factual realm a little bit and kind of getting a little more speculative, but I think this is kind of the point of this whole hierarchy, and also the point of this whole cortical circuit is to kind of combine this information, both the high level and the kind of low level information that we're actually getting, and use it to kind of build up our perception of the outside world. And I think this is kind of profound in a way, because our perception of the world is not actually what's coming into our senses, but it's actually something our brain actively builds. And what we consciously experience is not so much what's coming in, but the model that our brain has built. So I'm going to give you another example of this. So I have a little clip here of what sounds like R2-D2 saying something. So I want you to try to figure out what the message is. I'm going to play it one more time. Okay, so is anyone actually able to hear what it said? Okay, one or two people. You guys are good, but most people can't. Now I'm going to give you a hint as to what R2-D2 there was saying, the steady drip, or you can read it. So it's a little morose. I didn't come up with this example. It's Sylvia Plath, maybe, I don't know. But now I'm going to play it again, and I want you to see if you hear anything different. The steady drip is worse than a drenching rain. So everyone hear it now? Okay. So this is interesting because not only are you, kind of abstractly recognizing that it's the same as the sentence I just showed you, but you are literally hearing it differently. You could point to, which word occurred at which time, and you would be right. And so what's happening is your top-down knowledge is changing your perception of the outside world. I think this is pretty fascinating. And I think it's one of the points, basically, of this neural circuit. So that, I've been kind of talking in terms of perception, but this, could also be useful for motor if you're kind of comparing, what you want your motor action to be with the feedback you're getting from the outside world. I mean, this kind of general comparator circuit seems to be doing something very important that allows humans to do all the kinds of, or other animals to do the kinds of cognition that we do. And so this is kind of a major goal of neuroscience, is to figure out how this all works, and certainly one of my major goals. And so very briefly, I just want to talk about the kind of implications of if we could figure out how the circuit works. First, I think it's going to have enormous implications for medicine. So, autism, epilepsy, schizophrenia, you name it, almost any neurological disease is somehow a dysfunction of this cortical circuit, whether it be, miswirings or what have you. And so, basically figuring out how to, at this point, it's like trying to fix a car when you don't know how it works. You can kind of remove parts and put stuff in, but you're probably not going to make it better. But if you actually know how it works, I think we'll get much closer to being able to find cures for neurological diseases. Computing and artificial intelligence. There are a lot of things that computers are much better at than we are. So, for example, multiplying large numbers together. I think a $5 calculator could probably beat pretty much anyone in this room. However, there are things that humans are much better at than computers. So, for example, recognizing objects. Your average 6-year-old can beat, your most powerful supercomputer with really good programmers, trying to write computer vision programs for it. So I think if we can, we probably already have the processing power to kind of do these kinds of computations that the neural circuits are doing. They are impressive, but they're kind of within realms that are, if we haven't reached already, we can reach soon. What we really need to understand is the architecture of the circuit and what it's doing. And once we do that, I think we should be able to build the kinds of circuits that allow things like computer vision and speech recognition to happen. And also, we tend to think of inputs as being sensory inputs just because that's what we have. But the circuit does seem to be pretty universal, and we can learn new tasks with it. For example, we learn how to read, even though our evolutionary precursors never did that. And so that you could probably put kind of new types of sense information into this neocortical circuit and have it kind of intelligently kind of make inferences about it. And last, kind of token, human understanding. I just think it's, like what makes us different from cavemen isn't just that we live longer and then we have cooler toys, but also that we kind of understand our place in the universe, that the Earth revolves around the sun, that we evolved from other creatures and so on. And so I think understanding the brain is very much part of, kind of the both fascination and humility of the human experience. So hopefully at this point I've motivated you as to why the neocortex is interesting and worth studying. And now I'm just going to talk briefly about some of my own research on this. And so as I mentioned, I'm kind of interested in studying how this kind of primary visual neocortical region is encoding information about the outside world. And this has been studied quite extensively for about 50 years or so, and it is probably the best understood region of the neocortex. So for example, it's known that neurons in this region will fire action potentials when lines cross as part of the receptive field at a certain angle. So what I wanted to do is actually not look so much at just the responses to the outside world, but see how those responses change depending on the state of the animal. And we actually had good reason to think that they would change. So very early on, so I think this was in the 1930s, Hans Berger invented these EEG electrodes that you can, put on your scalp and will kind of measure the gross neurological activity. So this is filtered over, millions of neurons. But you can kind of look like very crudely at what's going on in the human brain during different behavioral states. And so this is from one of his patients. So on the top, you can see when the patient was excited, you see this very kind of low amplitude, high frequency activity. When he was relaxed, you see this kind of higher amplitude, slow frequency activity. And when it's asleep, you see like this very slow kind of high amplitude activity. Now, we know kind of intuitively that we kind of perceive the outside world very differently in these different states. When we're excited, we kind of pay attention more than when we're drowsy, and we certainly pay attention much more than when we're asleep. And we also know that this kind of gross neurological activity is going to affect the neurons in these neocortical circuits, and will likely change the responses. So what I wanted to see is, what is the biological mechanism of, how are these changes happening, and what effect is it having on the neocortical circuit? And so to investigate this, I picked a system which I had a strong hint was involved, and this is the acetylcholine system. So it's subserved by this nucleus here in the basal forebrain called the nucleus basalis. So this little region here, it's a very small number of large cells that send acetylcholine basically all over the neocortex. And the reason I was interested in this nucleus is because it's known that if you stimulate it electrically, you basically change the state of the cortex. So this is an example here. So I recorded the EEG from a rat, and you see these big slow wave oscillations. And then I stimulate this nucleus, and now you see these much faster, lower amplitude oscillations. So exactly the sort of state change I showed you in the last slide. It's also known that this system is kind of active at different levels depending on the state of the animal. So when we're asleep, it's pretty much turned off. When we're awake, it kind of fires tonically at like a higher level. And then when we become really excited, it kind of goes gangbusters and goes crazy. It's also known to be very important. If you damage these neurons, you have major problems with perception, attention, memory. So essentially, a lot of the pathology from Alzheimer's and senior dementia is thought to come from damage to these neurons. And finally, it's known that if you just kind of drop acetylcholine onto the visual cortex, it changes the response properties of the neurons. So what I wanted to do is kind of activate this area while measuring responses in the visual cortex to a natural scene and see how activating this area kind of changed the responses to the natural scenes. And so the way I did that, I'm not going to get into all the details in the interest of time, but I used kind of a clever technique where we got these transgenic mice from a lab at Duke, and you can actually specifically activate just the neurons that release acetylcholine by using a certain wavelength of laser light. So we drop an optic fiber into the brain, activate these neurons, and cause the acetylcholine release all over the cortex. Then in the visual cortex, I wanted to really record from a whole population of neurons rather than just one. So we used this thing, it's called a polytrode, and it's basically a silicon chip that has regularly spaced iridium sites on it. So this is the probe here, and then these are the individual sites, so they're kind of in an array. And then what happens is, so this is just kind of an example of, there's some neurons in front of this chip. Every time one has one of its little electrical blips in action potential, you can see it as a kind of signature blip in the electrical activity. And each one, because they're in a different location, will activate different channels. So this one over here will kind of cause that one, or that pattern of responses. And from that information, we can use mathematical techniques to extract the firing properties of a bunch of individual neurons and kind of look at populations of neurons and see how their responses are changing. So I have a ton of data on this that I unfortunately don't have time to tell you about, but I'm just going to show you one quick result to kind of give you a flavor for it. So unfortunately, colors didn't quite come out, but so what I've done here is I'm just taking a look at individual neurons. So I have 3 individual neurons in different colors here. So let's just look at this top one now, so the top 2 panels. And what I've done is I played the same movie over and over again for this neuron and looked at its responses. And so basically each little hash mark you see is a response to somewhere in that movie. And what I've done is I've stacked 30 responses to the 30 repeats of the movie on top of each other. And the reason why I've done this is that you'd expect that if the neuron is kind of giving you some useful information about the movie, it's going to respond at the same time during every presentation. Otherwise, if it's responding at different times, then it's not conveying anything useful to any upstream regions. And so what I wanted to see was, does the release of the acetylcholine actually kind of increase the information and the response of the neurons? And so what you can see is that if you look at the bottom, so the top 2 panels is the cell I'm going to show you. If you look at the bottom of those 2 panels, that's the response when I cause the acetylcholine release. And you can see that there's a couple of time points at which there's very reliable responses to the natural movie. But if you look in the control situation where I didn't induce the acetylcholine release, so the panel above it, you no longer see those kind of reliable responses. So it seems like what's happening, and you know, this is just one example cell, but we ran all the statistics. It's a very reliable effect. So it seems like what's happening is that the acetylcholine release is causing the cells to kind of better encode the visual environment. And as a kind of more quantitative way of testing this, what we did is we took the whole population of responses to the movies, and we tried to predict what movie or what part of the movie, which frame of the movie the animal was seeing, based purely on the responses. And then we found when there was acetylcholine present, we were able to do it with much higher accuracy than when there wasn't. So essentially, what's happening is the acetylcholine is improving the signal processing in that region. And this is really cool because what it suggests is that our brain, it's not like a silicon circuit where it kind of works the same way each time unless it's broken. It can actually change the mode in which it operates depending on kind of the needs of the organism. So when you're kind of zoning out, you're kind of not paying attention to the outside world because you're doing something else, and then someone snaps your attention, you release acetylcholine, and suddenly you hear what they were saying to you, which you were kind of ignoring a second before, even though it's the same stimulus. Just to sum everything up, so today I talked about how the neocortex makes up the majority of the human brain and kind of underlies our complex behavior, motor planning, cognition, et cetera. And that the entire neocortical sheet is kind of composed of a somewhat stereotyped circuit. And figuring out how this circuit works will be essential in terms of, in terms of understanding how the brain works. It makes up 75% of the human brain. And will also probably yield numerous practical benefits. And finally, I showed you kind of one example of how the neocortex is kind of a different animal from some of the circuits that we're used to. It can change how it processes information depending on the state of the animal. But I'm actually, despite these kinds of things which make it seem like it's going to be a very complicated problem, I'm very optimistic that we're going to be able to solve this problem, hopefully, within our lifetime. So there's a lot of new tools for, basically, so like what I did, recording from large populations of neurons. There's ways in which you can image the activity of large populations of neurons. We also have new tools which allow you to stimulate very certain neurons and not others. So we basically have all the tools now to kind of dissect parts of the circuit and really kind of take it apart and see how it works. So finally, I just wanted to thank especially my advisor, Yang Dan, and other members in my lab, and Guoping Feng, professor. He just moved from Duke to MIT, who he provided the transgenic mice we use. And I didn't really talk about the work, but these other people kind of helped me with various parts of getting the techniques to work. And finally, a big thanks to Peter for organizing this. He's been awesome. And so I think before we take questions, I'm going to hand it over to Emily, and she's going to give a short talk on her work. And then afterwards, we can do some Q&A.
Yeah, answer a few questions while I set up.
Okay.
Any quick questions?
Is that the chemical that the chemical
Acetylcholine?
is there any studies that show that your brain has more of that, like while you're performing at high, like intellectually or physically or like in an athletic sense? Is that something that's related to higher performance in general, or?
It's definitely thought to be related to higher performance and levels of arousal. Fortunately, it hasn't been studied carefully in humans. It's mostly been animal studies in which it's a little more difficult to quantify the amount of arousal. But it seems like there's been studies in which they found that if the animal's performing some task, it basically becomes, the nucleus releases acetylcholine during important parts of the task.
Thanks.
Okay, and hopefully there'll be time for more Q&A after. So I'm going to talk about something pretty different. I think that's one of the joys of doing a tag team talk, is that you can see the breadth of neuroscience research. We're both at Berkeley, but in pretty different areas. So I'll begin by saying that the study of neuroscience is devoted to understanding how the brain functions uniformly across members of a species. For example, Mike explored the way systems neuroscientists are tackling the profound problem of how cortex works. But a critical question is how neural and cognitive processes differ between members of a species, or even within an individual in different environmental conditions. Thus, the question becomes not how are our brains similar, but how do we differ? Thus, the purpose of this talk is to introduce you to two factors that contribute to individual differences in cognition, that is, genes and hormones. And because I study estrogen, apologies to the men in the room, because a discussion of the female menstrual cycle is imminent. I don't want to get too far afield, but I will say that a second loftier goal of this talk, and really this research in general, is to stress the importance of considering the role of hormones and sex differences in cognition. From a basic science perspective, an intimate understanding of how our endocrine system impacts neural and cognitive processes is interesting. From a woman's health perspective, I'll argue it's fundamental. The estrogen cognition literature, and in particular I'm thinking of the big population health studies on the use of hormone replacement therapy, that data is famously inconsistent. And we can ask, at the level of the brain, why? So before we start thinking about genes and hormones, we should talk a little bit about neurochemistry. Mike was talking about acetylcholine. I'm going to talk about dopamine. I assume many of you have heard of dopamine somehow in the room. So, like, in what context? You shook your head. What is?
Correlating with pleasure, motivation.
Exactly. Yeah.
Reward.
Yeah, Schultz is great. Yeah, so the reward pathway. Okay, that's good. So it turns out that there are three main ascending dopaminergic pathways in the brain. So you pointed out one, which is the mesolimbic pathway. So it starts here in the VTA or ventral tegmental area and projects to the limbic regions, the nucleus accumbens and such. There's two others. So the nigrostriatal pathway, which originates in the substantia nigra, another group of cells in the midbrain, and projects to the striatum. When the cells in the substantia nigra die, you get Parkinson's disease. And finally, the pathway in red is the mesocortical pathway. It also originates in the VTA. This projects to the frontal lobe, so prefrontal cortex, a part of the brain that's really important for things like planning and logical thinking and abstract reasoning, as Mike talked about. And that's the pathway that I'm talking about today. They're distinct pathways and they carry distinct cortical purposes. Now, it's been just over 50 years since Arvid Carlsson first defined dopamine as a neurotransmitter. At the time, in the late 1950s, it was thought to be merely a precursor in the biosynthetic pathway of norepinephrine, another neurotransmitter. His basic science work was really focused on the nigrostriatal pathway, and his work led to the development of using dopamine drugs to treat Parkinson's disease. He was awarded a Nobel for that in 2000. Really great work. But fast forward 20 years from the late 1950s to the late 1970s, and we meet Tom Brzezowski, who really demonstrated for the first time the functional importance of this red trace, the mesocortical pathway. And in a now landmark study, he was able to remove dopamine from the prefrontal cortex using this neurotoxin 6-hydroxydopamine. And he tested these monkeys on a short-term memory task, pre and post-op. And here's what he found. So in this task, monkeys are asked to maintain a bit of information over a delay period. In some trials, the delay is really short, and in others, it's really long. So the intact animals is represented by this white trace, and you can see there's this very expected or predicted decrease in performance as the delay period lengthens. Again, to be expected given the nature of the task. But what happens when we remove dopamine from the prefrontal cortex? These monkeys in the black trace, performance begins to tank almost immediately. What's striking is that the deficit seen in dopamine-depleted animals is almost as bad as those monkeys who had direct structural ablation to the prefrontal cortex. So literally, morphologically, there's damage to that region of the brain, which is shown here. And importantly, when dopamine was added back into the depleted animals, the effect went away. So the moral of the story is that dopamine is really important for proper functioning of the prefrontal cortex. This region of the brain is exquisitely sensitive to its neurochemical environment. So you modulate dopamine, you're going to modulate cognitive functions that rely on that region of the brain. So the general relationship between dopamine and prefrontal cortex is clear. I've given you one example, and there's a whole 50 years worth of work to show you more. But the intricacies of this association are not straightforward. So if I were to give everyone in this room 30 milligrams of methylphenidate, Ritalin, a common drug, I may see a quarter of you show cognitive improvement on particular tasks, a quarter of you show sort of a cognitive impairment, and in half of you maybe a null effect. That's interesting, right? Why is that? Well, it turns out that the effects of dopamine on performance are not linear. In fact, there's this inverted U-shaped function or parabolic curve that defines dopamine's actions in the prefrontal cortex. Now, this can be seen at the single unit level. If we're looking at the spatial tuning of prefrontal neurons engaged in a working memory task, we see that at optimal levels of dopamine stimulation, we get really good spatial tuning. But if we have too little dopamine receptor stimulation or excessive stimulation, we see this effect be degraded. Now, importantly, this is also seen at the human cognitive level. So just at the measure of behavior or neural efficiency, dopamine works in this inverted U-shaped curve. So there's this theoretical optimal amount at which you get the best performance, and on either end, performance begins to tank. So what becomes critical is taking into account this law of initial value, or where does an individual lie on this curve? Only knowing that can we predict how additional dopamine modulation is going to affect your performance, right? If you're starting out here on the curve, then you benefit from a dopamine boost, from that morning coffee, or even working well under stress, both of which are dopamine agonists. But if you're already optimal, then you don't want any modulation. So the natural question is, what does influence dopamine? Well, I already gave up the ghost and said genes and hormones, in particular estrogen, but also things like stress and coffee, drugs of abuse like cocaine, and prescription drugs like Ritalin. So I'm going to focus on the first two in this talk. So first, a quick note about the gene story. And to kind of approach that topic, we need to first zoom into the level of the synapse and understand dopamine's biosynthetic and metabolic pathways. So dopamine is produced in the synapse, and it gets released after. So Mike talked about the action potential, and we know that neurons communicate via electrochemical signaling. So there's the flood of ions down the axon, and then there's a release of chemicals, which, as you know from your intro neuroscience, that cells don't actually connect. There's a small space in between, and it's the job of neurotransmitters to relay that information. Well, so here's the instance of dopamine. It's being relayed in or released into the synaptic cleft. But there has to be some way for it to get removed after it's done its job. And it turns out in prefrontal cortex, the job of removing dopamine falls to an enzyme called catechol O methyltransferase, or COMT. And it's really important. So in the prefrontal cortex, COMT accounts for over 60% of all dopamine turnover. If we look at mice who don't express COMT, they show increased prefrontal dopamine and enhanced memory. Similarly, in humans, we can use particular drugs that inhibit COMT activity, and we see increased evoked prefrontal dopamine and enhanced working memory. Now, just a side note that in those other dopamine pathways that I showed you, the primary mechanism for dopamine degradation is something totally different. It falls to the something called the dopamine or DAT, a dopamine transporter protein. So this COMT enzyme is really particularly crucial for that mesocortical pathway for the frontal lobe. And that's interesting because the gene that codes for that enzyme is polymorphic. So there's a naturally occurring difference in the way that gene is structured in everyone in this room. So it's a single nucleotide polymorphism. What that means is that one of the base pairs is switched, and it's a guanine to adenosine flip. And this is very evolutionarily recent. So this polymorphism is first observed in humans. All animals have the G, the guanine, which is that base pair difference leads to the amino acid shift of valine to methionine. And the point I want to make is that sort of flip fundamentally alters the way the protein works. So in an individual, so about a quarter of the population who have this 2 copies of this VAL gene, it's an issue of thermostability. So that enzyme is physiologically under normal human body temperatures, it's very active. So it clears dopamine from the synaptic cleft really well. And we kind of colloquially refer to these people as having low baseline dopamine. So there's not a lot that's left hanging out in the synaptic cleft. The more recent allele, again, first observed in humans, that enzyme is actually thermolabile. So it begins to break down. It doesn't do a great job. And so dopamine's allowed to accumulate in the cleft, and we sort of refer to these people as having increased baseline prefrontal dopamine levels. And there's functional consequences. So VAL individuals actually have worse performance on these working memory or short-term memory type tasks and reduced neural efficiency, whereas MET subjects have better memory and better efficiency in terms of neural response. So, yeah.
Just in general or after receiving more dopamine?
In general. So just if we were to split this population into VAL and MET, we could see differences emerge without tweaking the system at all. So it's interesting. And this line of work is, at a big picture level, we can see that variation in genes leads to variation in cognition, but clearly there are many steps in between. So we go from a genetic variation, altering how a protein works, to altering how some neural circuit works, and maybe that bubbles up to the level of behavior. Maybe it doesn't. So the point of what we do, or as human cognitive neuroscientists, is to try to look at these endophenotypes or intermediate phenotypes. And we can probe brain function to see how genes affect brain function and maybe behavior. But we can kind of get like an early biomarker of how that gene is having an effect on the brain. Okay, so I'm going to skip quickly to the realm of endocrinology. In a breath, estrogen amplifies dopamine transmission. There's strong evidence in animals to suggest that estrogen enhances dopamine synthesis, release, and turnover, and modifies basal firing rates of dopaminergic neurons. We know that stimulated dopamine release actually fluctuates in a rat's estrous cycle. And finally, the PFC has amongst the highest concentration of estrogen receptors in the human brain. And hormones fluctuate throughout the menstrual cycle in this very stereotypical predicted way. So estrogen levels are lowest in the first few days of the cycle when the woman is menstruating, and it spikes to about 4 times the typical value right before ovulation, so around day 11 to 12 of the menstrual cycle. So given this fluctuation of estrogen during the menstrual cycle, and given estrogen's ability to promote dopamine transmission, here's what we predicted. If we could get some index of where an individual lies on that inverted U-shaped curve, we could predict whether estrogen was going to be helpful or harmful. So in order to do that study, and a series of studies actually, this is kind of a schematic of the experiment. We pre-screened a whole bunch of subjects for this comped enzyme. It's spitting into a cup, very simple. And then we teased apart the two extreme groups, so the Val-Vals and the Met-Mets, the low dopamine and the high dopamine. And then we brought them into the lab, and we gave them, we tested them on 2 occasions, so when estrogen levels are naturally low, when estrogen levels are naturally high, and on each occasion we gave them a battery of cognitive tasks. We gave them a functional magnetic resonance imaging scanning while they're performing different cognitive tasks, and we took blood samples for biomarkers. And this was the prediction that Val-Val subjects, we know from previous work, they're thought to have decreased prefrontal dopamine and thus reduced performance on these very highly dopamine-sensitive tasks, whereas Met subjects have optimal dopamine and really robust performance. Now, if we tweak that system and we say, if we consider this estrogen-induced dopamine shift, that we can get these subjects to be more optimal, to look like the Met subjects if estrogen levels are high. But in the Met subjects, we'll actually see the opposite effect, that their performance is best when estrogen levels are low, and they kind of overdose. The dopamine gets pushed over the edge under high estrogen conditions. So I've kind of alluded to cognitive task and prefrontal kind of mechanisms, but I haven't really talked about the kinds of tasks that we do, and I'm not going to give an example, specific example of what we do in the scanner because it's sort of long and boring, but I'll give you a kind of back of the envelope test of what we use in the field to test working memory or short-term memory. Okay, so I'm gonna give you a string of numbers, and I want you to repeat them back to me, or you can do it in your head backwards, okay? So here are the numbers: 2, 1, 7, 3, 4, 8. Is this what you said? 8, 4, 3, 7, 1, 2. Great. So that's working memory. You have to keep information in mind over a short period of time and maybe manipulate it in some way. And that's a gateway cognitive function for almost anything you do, certainly on a daily basis here at Google, right? So reading a paper and trying to relate one sentence to the next, doing math calculations in your head, that's all reliant on this really basic cognitive ability. And it's highly sensitive to the amount of dopamine you have in prefrontal cortex. So I'm going to cut right to the chase and show you a couple data slides. The first one is just behavior. So how do women perform on this type of working memory task? Well, it turns out that if you look at Val-Val women, so low, just naturally low prefrontal dopamine, so in the red bar, performance is about 75%. Not great. But if we look at these same women when estrogen levels are high, so right before ovulation, we see a boost in performance. Now, the interesting thing is the exact opposite effect is seen in Met subjects who already have optimal dopamine. Their performances peak when estrogen levels are low, which is most of the cycle, but actually performance goes down when estrogen levels are at the highest value. And to us, that seemed to be a suggestion that this inverted U dopamine response was actually visible in humans, taking into account estrogen and genotype. Now, I talked a lot about, you can run a gel and you can measure somebody's genotype for this comped gene, and you're either Val or Met or you're heterozygous. But what happens if we cut, sort of leapfrog over DNA altogether and go straight to the source, straight to the enzyme? And we can measure that. We can take blood samples. It's a little bit more invasive, but we can measure the active enzyme. And we do that because even within genotypic groups, there's a lot of variance. And that's a whole other discussion of why that happens, but it's really fascinating. The point is that if this model is correct, then I should be able to take into account this variance and predict at an individual level who's going to benefit and who's going to suffer. And the point of this graph is to show that even at the individual level, we can see this inverted U-shaped curve pop out by considering not only estrogen, but active enzyme levels at the time of FMRI scanning. So now on to the brain. So again, we did this in the context of a functional magnetic resonance imaging, or FMRI, study. And the data that I'm just going to show you is something called a region of interest analysis. So we know exactly what part of the brain is really important for working memory. So we can essentially draw a circle around that region. It's a task-related region of the brain, and just look at activity, mean activity across that region, and compare it between groups. And essentially what we find is a representation of that behavioral effect, but in terms of neural activity, or bold, fMRI bold response, such that the less optimal dopamine groups show a sort of weak effect in this part of the brain, whereas the more optimal dopamine groups show a really robust effect in this brain while you're performing the task. And importantly, the extent to which any individual shows this nice, robust activation during the task was predictive of their performance. So we see that dopamine status predicts PFC activity, which in turn predicts an individual's performance on this working memory task. So in summary, estrogen improves working memory in women with genetically low prefrontal dopamine, but impairs performance in women with naturally high prefrontal dopamine. And we see that this inverted U dopamine effect is reflected in behavioral and neural indices of PFC function. So a few implications. First, dopamine's influence on cognition cannot be fully understood without taking estrogen into account, I argue. Second, the functional impact of genetic variation can be explored via direct assays of brain function. And finally, this kind of loose concept of women's health can be addressed very specifically within the central nervous system. And there's actually a really great edition in Scientific American Mind that's talking all about this his and her brain. Okay, and a final slide. Why should we care? I think Mike and I both get our armchair points of talking about this at the end. Often in neuroscience, sex is something to be controlled for. Or scientists leapfrog over the matter altogether by studying only males of a species. To make that point clear, colleagues at Berkeley recently indexed all studies in mammals published in 2009 across 10 fields and over 40 journals, and they found a widespread bias towards using only male animals. The effect was especially striking in neuroscience, where the ratio of male-only to female-only studies was 5.5 to 1. Now, there's a logic to that. It's cheaper. You don't have to track things like females' menstrual cycle, so there is some rationale to that. But the interesting thing is that this bias was largest in neuroscience, pharmacology, and physiology, fields for which the basic science research has huge implications for human health. If we only study male species, we're going to miss out on a few things, is what I'm trying to argue. Sex differences are repeatedly found for rates of depression, vulnerability to stress, pain, Parkinson's disease, ADHD, and nearly all aspects of drug abuse, from acquisition to treatment. We know that enormous endocrine changes happen during menopause, but we know relatively little from a human neuroscience perspective about how this normal aging process affects specific neurochemical and cognitive systems. According to the news, one day estrogen is good for you and the next day it's bad for you. So yes, it's a messy problem. I think the good ones always are. But the problem gets thrown into relief only after it's subjected to careful scientific study. So hopefully we'll continue to do that and understand a little bit more. But in general, hormones and genes play an interesting role in terms of brain function. I hope I've convinced you of that today. Thank you.
Emily, Mike, thank you for fantastic and really exciting perspective on things. I'd just like to open up the floor to questions, and I'll extend it to the patient people on VC first, just to see, does anyone on VC have a question? Last chance? All right, I'll come back to you. Anyone in the room?
Thanks both. Those were both really great talks. I have a question for Michael first. It looks like you used optogenetics to stimulate the acetylcholine.
Yeah, exactly.
So that has really great timing patterns. Have you guys looked at all of that, the effects of when you stimulate? Does it really matter?
So with the acetylcholine release, it works a little different. So usually timing is very important for neurons, and the exact kind of timing of the stimulation you give them really affects how they fire. Acetylcholine is a little different in that respect, in that it's when the neurons release acetylcholine, they release it in this kind of bulk way. It goes all over kind of extracellular space, and it takes a long time to clear. So the effect is much more kind of, it's like spread out in time, basically. And so I looked a little, basically what I found is when the light is on, the effect is there. When I turn it off, it goes away relatively quickly. It's not instantaneous. So giving shorter pulses, it would tend just not to activate it at all. I kind of had to give like a longer continuous pulse.
One other question. So it looks like you record in V1 to see the effect. Did you have a particular hypothesis it should be there? Do you expect it to be in most parts of the brain?
It's probably going to be all over the or at least the sensory neocortex, and it also projects to the prefrontal cortex, but it's just we don't really know what's going on there. I picked the visual cortex just because we have pretty good models of what's happening in terms of sensory processing already, so it makes it much easier to interpret the results.
Thanks.
Can you talk a little bit about the heterozygotes, or is it dominant behavior, or what are you seeing?
Yeah, so in my study, I didn't look at the heterozygotes, which represent 50% of the population, because I wanted to look at the extremes and see the effect. But in studies that do include them, you find basically that they fall right down the middle. So it's interesting because there's another group led by Danny Weinberger at the NIH who studies the effects of drugs. And you can actually, in pharmacogenomic studies, you can predict the effect of a dopamine drug. He's used, I believe, methylphenidate and amphetamine. You can predict the effect of that drug based on comp genotype. So you see improved both neural efficiency in terms of FMRI bold and also improved performance in val-val subjects, slightly improved performance in heterozygotes, and actually impaired performance in met-met subjects. Super interesting. I mean, it speaks to the fact that we, at least in theory, could begin to do individualized medicine, and this is kind of one example.
I was wondering, for people who had too much dopamine, did you notice increased anxiety and other things like that?
So I measured sort of state and trait anxiety levels, so during the day of the scan, because we really wanted to control for that. Now, it's an interesting point you bring up, because people say, well, now, obviously, Mets are the best, right? They have better working memory, like we should just figure out who they are and hire them all at Google or something. But it's not quite that simple. I mean, the reason we have both genotypes is, it could be vestigial effect, but in fact, Met subjects have increased rates of anxiety. So it's hard to say that, there's a better or worse. It's just different. It depends on what your measuring stick is.
Did you look at serotonin as well as dopamine?
I didn't, but that would be fascinating. Next 5 years of my life.
Just a simple question. What's the prevalence of the two alleles, the MET and VAL?
Yeah, so they're codominant. So about 25% of the population are VAL-VAL, about 25% are MET-MET. In my sample, it was slightly less. I think I had about 20% were MET-MET. And again, 50% are heterozygotes. And yes, of course, I genotype myself. So when my estrogen levels are high, forget about it.
Question for Michael. You were talking about how the brain effectively feeds sort of top-down abstract concepts into, say, visual perception. Now that's something obviously that computers lack, so we're going to struggle to try to get a computer to have these same sorts of things. I presume that sort of develops during growth as a baby. It's sort of like the brain bootstraps these concepts to a higher level.
Yeah, so it's not known exactly how that happens. What seems to happen is that the cortex kind of develops hierarchically, kind of moving up. So like the earliest areas develop first, so like the primary visual cortex, for example, that first level of visual cortex. It's very early in development when it kind of 2 to 3 years of age in a human, while the prefrontal cortex is still kind of developing. Even teenagers, kind of early 20s, are still kind of developing their prefrontal cortex. So it seems like the cortex is kind of getting progressively trained, like in a hierarchical fashion.
Okay. What do you think computer vision would benefit from trying to sort of do the same sort of bootstrapping, perhaps even force the top-level concepts in to begin with?
Yes, I think it could be. So, I mean, in some ways, it's a very different strategy of computing, and I think for doing the types of things that computers are currently good at, I think it's not going to be very useful. But for doing kinds of things that humans are good at, I think it will be very useful. So I think it will almost be like a different type of computer. Totally speculating here, but I think it will be good at the kinds of like object recognition type tasks, and less good at kind of serial, straightforward tasks.
I have a quick question for both of you, but I'm having a really hard time resisting hating you guys for being both brilliant and beautiful people. So it's with all of these findings, we get closer and closer to larger questions of can we control and should we control these things to enhance our own abilities. Perhaps there's a better way from just understanding our biorhythms, or perhaps healthier ways of addressing better releases for either dopamine or acetylcholine. But I was just curious from both of you what your perspectives are on those things.
So I think the first kind of priority is to figure out cases in which these systems are working incorrectly, in neurological diseases, and fix those first. Then comes the question of like, okay, we developed this, way of kind of manipulating the acetylcholine system in order to help people with Alzheimer's disease. Turns out you can kind of give yourself, a little boost on test days. Should you do it? That's kind of like a question for a bioethic. I mean, I don't know quite how to answer it. I guess I'd say yes under certain circumstances, but I think that the circumstances are tricky.
I guess I would just agree that, this is kind of the question that we always get, especially with the comp genotype. The important point is that, I talked today about one single nucleotide polymorphism, right? But there are, an infinite number in the brain, and in the neural regions, genes don't operate in isolation, right? They all work together, and they're part of this larger system. So, until we can understand what really the ultimate good is, like what is the best, it's not just MET genotype, it's MET genotype plus, D1 receptor polymorphism of it's like going to be a whole concert. And so we're just literally, scraping the top of the iceberg off when we ask these questions. So maybe that's a cop-out answer, because all scientists say we have to know more, whereas the general public says, where can I buy the drug? So at some point, we're going to have to confront these questions pretty soon.
I'll extend the invitation to VC. Anyone out there with a question? All right. Well, thank you so much. You've given us a lot to think about and be inspired by.
Thank you all for coming.